Welcome everybody. My name's Carl Larson from RMCG, part of the Soil Wealth and Integrated Crop Protection team. And today we're going to be covering managing cucurbit viruses for profitable vegetable production. Really today we wanted you to go away with some really practical tips and tools about how to manage these specific viruses on your farm or with your clients if you are an agronomist. And we're really lucky today to be joined by Craig Webster, who's a plant pathologist with the Department of Primary Industries and Regional Development. Before I do hand over to Craig shortly, just a little bit about Soil Wealth ICP. If you're not aware, it's a project centred around getting research onto farms around improving soil management and plant health in the Australian vegetable industry. It's funded through Hort Innovation using the vegetable levy and contributions from the Australian government, and RMCG and our partners AHR deliver that nationally and have for the last 7 or 8 years. If it is the first time that you've joined one of these sessions, you'll notice up on your screen there's the GoToWebinar control panel. A couple of things to highlight there. There's some handouts for you to click and download directly without having to navigate through websites that are relevant to the topic today. And there's also a spot there for typing in questions, which will come straight through to me, and then we can put those to Craig at the end of his presentation. So without further ado, Craig, welcome, and I'll hand over to you and we can see your slides bright and clear.
All right, great, thank you. Yeah, so as Carl introduced, this is sort of work we've been doing the last couple of years on cucurbit viruses through the Hort Innovation funded area-wide management project dealing with viruses and bacterial diseases. So this is a summary of the sort of cucurbit viruses, what they look like, a little bit of what they don't look like, and how they get about, where they survive, and some of the steps you can take to manage them for more profitable vegetable production. Doesn't want to move slides. So the talk, I'll sort of split it up into two main sections. We've got one group of viruses I'll term cucurbit potty viruses. This is the sort of family that they belong to, but zucchini yellow mosaic, watermelon mosaic, and papaya ring spot virus. We've found them across Australia, but we typically find them where zucchini yellow mosaic is in Western Australia most years, ZYMV maybe around Sydney Basin quite a bit or several years, and papaya ring spots more of an issue in Queensland, but more or less they're relatives of each other, and the way they survive and spread is pretty similar, and so is the management of them. And the second one obviously was cucumber green mild mottle mosaic virus, CGMMV. This, it is in many states around Australia, but in most places it's sort of restricted to fewer farms, whereas the cucurbit ones might be present in most areas in a district. This maybe is on a couple of properties or perhaps none at the moment, but we sort of picked these two as they're the two main ones we see around Australia, and they're very rapidly spreading in sort of bad years and can really quickly spread to a whole crop and cause a lot of damage. So that's kind of where we've looked at these two. There's something everyone has to contend with, one or the other of them, so it's an issue for lots of growers. And just a sort of a summary, so we've sort of started creating some guides from this project. There'll be a virus guide out a bit later this year. It's with the printers, but just to give you a rough idea, the way that they spread is quite a bit different. Obviously, the spread with winged insects like aphids, some of the chewing insects maybe over this side. Seed, particularly as a sort of primary source of introduction to CGMMV, is pretty important. Both of them can survive in crop debris. Weeds, these guys are probably a little bit more at risk for, but we'll just sort of go through, you know, how these guys are sort of spreading and where they're coming from, and give you a couple ideas of management for them as well. So first of all, these three viruses, ZYMV are the particular examples here, but they're fairly indistinguishable between the three of them. You're not going to be able to point in a field and go, that's definitely virus ZYMV and it's not WMV. We all have doubt, even myself and other people working on it for decades, because it is really dependent on what cucurbit type you are, what variety, what virus, how long, the weather. So you can get a general idea from these where they cause this kind of, in this middle photo here, we've got some rock melon leaves with the vein yellowing. That's something that's more kind of particular in a rock melon for these viruses. A little bit on the pumpkin on the right here as well, but they'll start off on these younger leaves, and then as the plant continues growing, you'll start to get really severe symptoms, shoestringing, where they get kind of stretched almost, and this mosaic like patterning of light and dark green areas on leaves. And it's typically on those young leaves that show up, and that kind of distinguishes it from more of the nutritional disorders or deficiencies, where they maybe show up on the lower leaves, and you usually get dark green veins and sort of yellow leaf panels instead. So this is the sort of reverse of that. And all of these will affect fruit and the leaves a little bit depending on the virus and the crop that you're working with. Some of the fruit symptoms, some of the more severe ones, we've got a zucchini here on the bottom left. Obviously, you can see that blistering. It's almost kind of raised, blistered areas on it, and dark and light green. Some of these other, the watermelon here on the top right, is fairly distorted and very strong patterning as well with it. But you can get milder symptoms here, like this watermelon with just very faint halo, kind of yellow depressed spots. So it's not so striking in the field, but it can affect the fruit texture and flavor and sugar content. So you still have to be wary of those. And generally the rule is that the later the plant gets it, the more the fruit have started growing, the milder the symptoms will be. But just depends on markets whether they would tolerate that kind of problem, or if yields are supply is short, then those might be acceptable still. Butternut here as well, you can just see very faint in the middle, very faint green circles. So those kind of things is what we see, and there's not many other disorders that look quite like those. A few that do, just to try and give you some examples. For instance, here we had some herbicide damage on some cucumber leaves, and just the way that the whole leaf panel in some areas was sort of bleached rather than yellow was a sort of giveaway that it was more of a herbicide damage. And you can also see if it maybe is on a row edge or something, and there's been weeds sprayed off recently, stuff like that. Mites and other insects can often be a bit confused. I've got some in the capsicum here. I haven't got too many examples in cucurbits, but spider mites are usually pretty obvious with the webbing, but broad mites, usually if we get samples, people concerned about viruses, and it's not, it'll be something like a broad mite. So a hand lens or maybe a macro lens on a phone can kind of give you an idea to check the back of the leaves, and if you see these kind of insects running around on the back, you can see if there may be mite damage instead. And the other one we had a few examples of coming through the project was people worried about this brown etching on some pumpkins, but it's really much more of a physiological fungal pathogen complex. It's not caused by viruses, so that at least is one, but it is very striking. So the circles are very kind of characteristic of viruses, but this one unfortunately is a different issue. So how do these guys spread? The three of them really all spread from weeds, such as this kind of cucurbit weed called a mukia growing out on a fence line. Volunteers, we've got one here where there's a crop last year. Obviously fruit was discarded. They might have had virus, they would have had virus issues because this small plant in the foreground has kind of germinated. The whole plant is virus infected, and it's a really potent source of spread into some of the older crops which have been planted a few meters away. And usually we say with these volunteers and weeds, within about 50 meters of crops for these cucurbit polyviruses is the ones that we've seen spreading into crops pretty efficiently. They're spread by these little guys, the aphids. And there's a lot of different types of aphids out there. The ones we think of as colonizing that start kind of breeding in cucurbit crops is usually the melon or cotton aphid. But it's really important to note that the aphids that don't breed in cucurbit crops, like ones that live on grasses or solanaceous crops, those guys are all can be quite effective vectors of that virus as well. So you won't necessarily see them breeding in the crop, but as they fly over those crops, they can pick up and spread virus pretty rapidly. We call them non-persistent because they can only do it for a short period of time, but they only really require a few seconds to feed on an infected plant and then on a healthy plant to spread that virus. So those guys that don't colonize, they may only spend a few minutes in the crop, but that's more than enough time for them to pick up and spread virus. And this real quick turnover of virus by aphids can make it really hard to control them just by insecticides, because while insecticides may eventually kill them, they've ingested enough to die, it won't kill them fast enough for them to have picked up and spread that virus. And in certain cases, the insecticides make their behavior more likely to spread it, because they don't settle as much on a single plant. They will move to more plants. So that's the first bit maybe of management. It's a holistic approach for these viruses. Unfortunately, one thing like insecticides isn't going to work. So roguing of symptomatic plants, there is some spread of these from leaf to leaf. So usually if they're sort of intertwined with their neighbors, we recommend getting rid of them. If they're quite clearly independent, then a single plant is enough. This really only works maybe when there's only a few symptomatic plants in a crop. You wouldn't want to do it when there's half the crop affected. It's a bit late by that point to stop further spread. It also takes maybe 1 or 2 weeks for symptoms to show up. So if half of them have it, it's likely the other half have a pretty good chance of having got it already. They just don't show it. The 3 weeds we deal with mostly, Mukia, paddy melon, and Afghan melon, they're all cucurbit weeds. They're fairly common in lots of different systems, so they're reasonably common weeds around Australia, and they're also fairly mildly affected if symptomatic at all. So they can often be really close to a crop and they look fine, but they have virus in them. We just can't see it particularly clearly. As I showed, I think on this bottom left photo, barrier crops is a really sort of different way of looking at controlling these, because while the virus is spread very quickly by the insects, it's also lost very quickly by them. So what we find is with a tall kind of barrier crop like this, particularly upwind of a zucchini or other cucurbit crop, once those aphids land on that plant material, and they're not very strong flyers, and they're very they like sort of just traveling along in the air and settling on plants as they come across them. Once they feed on that millet, which isn't a host of the virus, once they feed even for a few seconds, they'll have kind of cleaned their mouth parts out, and then they're no longer infectious. So if they do carry on over your crop, they won't actually have virus anymore to spread. So that's one way of looking at that. But I'd still say scouting in those first kind of rows to make sure you don't have the odd virus infected plant that get through would be a wise decision. Resistance is a really good mechanism usually for controlling viruses. Unfortunately, in cucurbits, only zucchinis have resistance that's very useful for these viruses. And we had some information out of the project. Cherie will have some more when we finish the final report up, but we looked at, in this particular field trial, we had resistant varieties which are marketed by a variety of different seed companies with this resistance to these 3 viruses, or 1 or 2 of them, and they still produce normal looking fruit compared to the susceptible varieties. And some of the older kind of industry standards like Eva and Rosa are in that kind of susceptible group. These ones had fewer symptoms, and where they did have symptoms, they were milder, and where we measured the amount of virus in them, there was a huge reduction in virus amount, which meant that they would also be less likely to spread to other plants in the field. And probably the last thing to mention, sorry, is about looking at vectors. Looking just on the leaves for the colonizing ones is sort of useful in some ways, but it does miss a lot of the information. So some of these ideas like sticky traps located out in fields, you can start to get an idea of when those vectors are flying through. and follow that up at that 1 to 2 weeks after a flight of aphids. When you first start noticing them, that's when those symptoms will start showing up on the cucurbits, and you can start looking at roguing them out to reduce them as sources of further spread. And you can also then see, maybe is there one edge where there's a virus and one corner perhaps, or something like that, and start to look at what weeds or volunteers may be nearby that you can get rid of those sources of virus, because it's very hard to control obviously aphids in an entire population, and where these guys, they just keep flying through, it's tricky to keep the crops free of aphids spreading virus. So I'll sort of change tacks now. This is the second main virus that we looked at, cucumber green mottle mosaic virus. In some ways, when we sort of write them down, what the leaves do, you can see, we'd say it's really hard to distinguish them from the 3 potty viruses we looked at before. I think here in the cucumbers, it looks a little bit nutritional almost, but some of these upper top leaves, it's still, for us with that kind of symptoms and vein yellowing, it almost looks like zucchini yellow mosaic virus. So it's very hard to tell them apart, and it's one of the reasons why correct diagnostics is important. The leaves and the fruit, again, are fairly obviously affected. Some of the cucumbers also, apart from just being, they might look fairly normal, but we hear reports of them being very soft and sort of squishy. So obviously their shelf life is reduced. Melon, watermelon are quite severely affected. Cucumber is bad. And here is that same mukia weed, which was infected and had no symptoms at all. And we also had a loofah, a sort of Asian vegetable, and there's a sort of leaf in the middle here that's just mildly kind of yellow, and that's about all we could see of symptoms. So often these other crops or other weeds can be pretty misleading and overlooked as sources. That's really where it hides out. It spreads predominantly by touching of infected and healthy plants, and it's very easy to do so. It's probably the easiest virus in plants that moves by touching alone. It copies itself to really, really high numbers in infected plants, so then the smallest amount of abrasion, and cucurbits are already pretty scratchy, lets the virus in, and it can spread really, really quickly from a single plant in a field to 100%. There's been a lot of talk in literature about the spread of it by some of the chewing insects and bees. I think that definitely can move it across into new blocks and be an issue of concern, but within a single field, the spread by touch happens so fast that it probably moves through a whole cucurbit crop before chewing insects can spread it. But if that block died off and all those chewing insects flew to a neighboring field, that could be part of how it spreads around on a farm. The other sort of difference to a lot of the other viruses is in cucurbits and other vegetables as well, is it's really contagious and can sort of up to maybe 2 or 3 years of survival in the soil. So if you have long rotations with other crops, it can still be there a couple of years later. I think that one really good way of reducing that risk is through trying to break down as much of that organic material as you can. Where it seems to survive the longest is where you have desiccated, dried out leaf material, and even things like maybe tendrils in a shade structure where they've sort of grasped onto something, and as you clean the crop out, there's some of those tendrils left. New crops coming and touching those sort of dried out, desiccated material is pretty infectious, whereas just on its own, kind of loose in the soil with all the other bacteria and fungi and things in the soil, it's not going to survive as long, but it does survive longer than most other viruses. So it's an area to look at. Plus, not just in soil, machinery, tractors, the rubber tires, boots, secateurs, people's skin, it can survive on those kind of surfaces for hours or days perhaps. Again, especially if you've got leaf material or something like that, that really does protect it and allow it to survive much longer. And so then finally it can survive as a volunteer, so it's seed-borne, and it can sort of come into crops if unclean seed is used from disreputable sources. And it does also infect some weeds as well. Mostly the cucurbits are the ones that it seems to have the biggest concentration in, and it's the biggest risk of getting back into cucurbit crops. So things like mukia, Afghan melon, paddy melon, stuff like that. So the management for this one, some of them are very simple. Again, we say removal of symptomatic plants, particularly neighbours, even if you're not sure if they've been touching, the roots may have underground that can spread that way. So get rid of the neighbouring plants, weeds, volunteers as well, and dispose of these away from crops. And depending on what options you've got, deep burial, burning, things where that virus infected material is not going to come back in contact with that crop or subsequent crops. Again, there's good resistance to this virus in cucumber, but the way it works in cucumber is that the virus still infects those plants. They just don't show symptoms anymore. So be aware of that if you then want to plant watermelons or some other cucurbit, that those tolerant cucumbers may be really good sources of virus to survive. And because they don't show a lot of symptoms, you won't realize there was a lot of virus in there. But if you're just growing cucumbers, it's less of an issue. And there are a lot of information and fact sheets out around disinfectants. So bleach or sodium hypochlorite is a really good one. Virkon quaternary ammonium compounds are some of the sort of detergent type compounds you can buy. They will all work to disinfect. They'll help to dislodge and break down any virus on boots or tools or something like that. But it isn't instant. It does require a sort of period of time for it to happen. And then you also need to make sure that if you've got, say, Virkon or something like that in a foot bath, that they can become saturated, so they do need replacing. You can't just leave a Virkon foot bath out for 6 months and expect it to still be working as well. And particularly as this isn't a virus present on lots of farms and it isn't as easily airborne spread through things like aphids, on-farm biosecurity, having visitors report to the office before they go on site, and you can check boot sprays and wash down areas for vehicles and stuff like that can really help. Even if neighbours do have it, it can help that coming onto your property as well. And lastly, as I said, these two things are really, really indistinguishable, CGMMV and the potty viruses. So knowing which one you're dealing with can be really helpful to start managing them, because you probably can't do all of these methods, but if you know which one it is, you can really start to chase in on which ones are important in that particular circumstance. And I think, yeah, Carl had some other fact sheets as well on this, but there are a few fact sheets specific on CGMMV in Vietnamese and in English. So you're welcome to go to the website and see if there's some information there that's useful for you, as well as information about biosecurity checklists. And I think I've got a few minutes left. One of the other questions we had a few times was around some phytoplasma diseases. They're quite different looking in cucurbits. They spread like viruses, but they're bacteria and they're kind of spread by some flying insects, but we rarely see them as an issue. So general sort of hygiene and removing and disposing of all crops without trying to touch them too much and dispose of them away from crops is smart, but in this case it's not a particular risk. But just if anyone has seen those, they're not actually caused by viruses. And I think that's it. Thanks. Probably run over time a bit, but happy to answer any questions now if anyone's got them.
Excellent, thank you, Craig. Some fantastic tips and tools in there, just walking through the ID, the symptoms and sources, and some of the management. Over to you now, our participants, for any burning questions that you had for Craig. Feel free to type those in the chat. As we wait for people to do that, one thing you mentioned, Craig, at the end of the CGMMV slide around management was the importance of accurate diagnostics. How do you best go about that? Where do you go? How do you prepare your sample and make sure you're dealing with the right thing? Yeah,
so I think it's through each of the states, we've obviously got our diagnostic services. We're DDLS in WA, but we have partner kind of companies or state government agencies running through those diagnostic services in other states. If you go to our website, we have information, even just contact details on who to ask locally to you. And also in the disease guides that Cherie is putting out as part of this project, there's information there on how to sample and what to sample. A couple of beginner mistakes sometimes: make sure it's kept cool as much as you can. Don't leave it in the back of a car for 5 days, it will rot. And also, if you're going to send it, it's good to send it with an ice block or a frozen water bottle, but wrap it up in some paper towel, because a leaf in an envelope or a box next to a frozen water bottle tends to sort of melt. It all breaks down, so it becomes pretty untestable after that. And yeah.
No, thanks for that, Craig. And we've got a question in from Michael just asking, what is the reason for CGMMV not spreading via aphids, but can or does spread via chewing insects?
So the viruses, the cucurbit viruses, it's a really kind of specific lock and key type mechanism that they interact with those aphids, and the chewing insects don't have that, so that's why those viruses don't spread by chewing insects. The CGMMV, because it's so mechanically stable and infectious, and it's like little squat rods, almost like little steel girders is its actual structure. And so those chewing insects, when they're chewing up on a leaf, their whole mouth gets kind of contaminated with it. It's just physical dirty mouth parts. And if they then fly off onto another healthy plant and chew on it a little bit, some of those viruses can get in because they wound them when they chew the cells. And as long as the cell gets a few viruses in it, but the whole plant doesn't die or the whole leaf doesn't die, then the virus can survive like that. That's the sort of thought of how they spread.
Thanks, Michael, and thanks, Craig, for that response as well. Another question we've had come in too is just around the importance of monitoring. How you do that, where do you start, and how that might differ from perhaps some of the more traditional scouting we do, say, for things like insect pests.
Yeah, I think with these guys, scouting with insect pests, you've obviously got a lot of different things that you're looking for, and yellow sticky traps is one of the ways to capture the most information that's relevant for virus transmission. So looking at those upwind borders where insects will fly in. Once you start to see, like I said, those aphids stuck on sticky traps, and particularly with things like hand lenses, you may not get to an exact species, but you can start to recognize the different orders of insects. You know that they've started flying through the location. You know, maybe 1 or 2 weeks later is that first risky period for virus to show up. So it's not about necessarily having to go out every week, every day. It's knowing those peak risk windows, and you can help to start pick them up with the sticky traps. And also, obviously, if you've had a really wet season locally, then you know that insects are likely to be in higher abundance, and you can start to find those windows opportunities earlier for them. And then with some of the management, like the cucurbit zucchini resistant variety lines, the growers we speak to, they do like the traditional varieties more. They prefer the fruit, the set, the number of the older varieties. But what we sort of found with some of them was in those high peak windows, if you looked at some of these newer varieties, you would get a decent yield from them, whereas those industry standards, you might get nothing if there was a lot of virus around. So you don't get everything, but you can get the most possible yield that you can in those particular windows of maximum risk.
Yeah, great. Thank you for that. Now, that was a very quick half-an-hour session, but
yeah,
the first in our area-wide management webinar mini-series. So we've tackled cucurbit viruses today. Early September, we're going to do a bit of a deep dive into a similar session, but for capsicums, and then in October, the same for lettuces. So we've got a 3-part mini-series. This was the first today. If there were any other burning questions, please feel free to send those through and we can deal with those offline. And if you haven't registered for the subsequent 2 sessions, jump onto the Soil Wealth website, soilwealth.com.au, and sign up for those and make sure it's in our diaries for the 7th of September and the 5th of October coming up. But Craig, thank you for joining us. It's been great to have you along, and we look forward to seeing everyone at the next session. Thanks, everybody.
Thank you.
Bye.
Bye.